Looking at this logarithmic pH scale, we can map the chemical gradient maintained between the systemic blood and the stomach. Every day, the stomach produces roughly 2 liters of gastric juice at a pH of approximately 1.0. Systemic blood, circulating just millimeters away, is regulated at 7.4. The fluid inside the lumen is over 1 million times more acidic than the blood supplying it. This high acidity serves 3 specific functions. It acts as a bactericidal barrier for innate immunity, it denatures dietary proteins to expose their peptide bonds, and it provides the environment required to convert inactive pepsinogen into the active proteolytic enzyme pepsin. This environment also facilitates the chemical reduction of dietary ferric iron into ferrous iron, which is necessary for absorption in the small intestine. Generating this fluid is the responsibility of a specialized cell located deep within the oxyntic glands of the stomach fundus, the pyramidal parietal cell. Pumping protons against a 1,000,000-to-1 gradient requires a substantial expenditure of metabolic energy and strict spatial segregation. To prevent self-digestion and metabolic failure, the parietal cell must operate as a perfectly polarized machine, separating its corrosive exports from its systemic exhaust. This cross-section maps the parietal cell, the apical boundary facing the stomach, and the basolateral boundary facing the bloodstream. Inside, carbon dioxide combines with water, forming carbonic acid. This rapidly dissociates into a proton and bicarbonate ion, a reaction accelerated by the enzyme carbonic anhydrase. To force this generated proton across the apical membrane into the acidic stomach lumen, the cell uses the H+K+ ATPase pump. This pump actively extrudes a proton into the lumen in direct exchange for importing a potassium ion into the cell. Because this primary active transport mechanism moves ions against a massive concentration gradient, it requires a continuous hydrolysis of ATP, making the parietal cell one of the most energy-demanding structures in the body. To maintain electroneutrality, chloride diffuses out of the cell through a passive channel on the apical surface, joining the proton in the lumen to form hydrochloric acid. The apical membrane functions strictly as the cell's energy-dependent exporter of acid. However, generating meeting protons leaves a dangerous byproduct behind, a surplus of bicarbonate. If this bicarbonate accumulates within the cytoplasm, it will trigger an intracellular alkalosis, disrupting the cell's metabolism. The cell solves this on the basolateral membrane using a chloride bicarbonate exchanger. This antiporter protects internal pH by expelling bicarbonate into the bloodstream while pulling a chloride ion inside. This inward movement supplies the chloride needed for the passive leak at the apical membrane. By dumping bicarbonate into the venous circulation during a meal, the stomach temporarily elevates blood pH, a measurable event called the alkaline tide. This systemic alkalinity is eventually neutralized when the pancreas extracts that bicarbonate to neutralize the acidic contents entering the small intestine. Gastric acid production is a balance transaction. For every proton dumped into the stomach, a bicarbonate ion must be deposited into the blood. To prevent continuous acid secretion, the secretory activity of the parietal cell requires tight regulatory control. Control is managed by 3 basolateral receptors, M3, CCK2, and H2. Binding acetylcholine or gastrin to M3 and CCK2 activates an intracellular cascade, releasing calcium to stimulate the pump. Meanwhile, histamine triggers the H2 receptor, elevating cyclic AMP as the most potent individual stimulant. Because these receptors use distinct second messenger pathways, they exhibit synergy. Activating all 3 yields an exponential increase in acid secretion that is greater than the sum of their individual effects. This synergy explains why H2 receptor antagonists, such as famotidine, cannot achieve complete acid suppression. Even with the histamine pathway blocked, the calcium pathways from acetylcholine and gastrin remain active and continue driving the pump. To stop acid production entirely, the pharmacological target must be shifted from the upstream receptors to the machinery itself. Proton pump inhibitors like omeprazole do this by irreversibly binding directly to the H+K+ ATPase. By disabling this final common effector, these drugs stop acid production regardless of how many stimulatory signals are being sent to the basolateral receptors. To understand how these signals are coordinated, we must trace the gastrin signal back to its source in the gastric antrum. Gastrin is secreted by specialized endocrine cells called G cells. They release the hormone into the circulation in response to mechanical distension of the stomach and the presence of amino acids. The vagus nerve directly innervates both the parietal cell and the G cell, but these two branches utilize different neurotransmitters. Vagal fibers release acetylcholine onto the parietal cell, but the fibers synapsing onto the G cell release gastrin-releasing peptide, or GRP. This chemical difference becomes critical when a patient is administered an anti-muscarinic drug like atropine. Atropine successfully blocks the muscarinic receptors on the parietal cell, but because the G cell synapse relies on GRP, the vagally mediated gastrin signal remains completely unobstructed. Presuming that the autonomic nervous system uses identical neurotransmitters at every synapse leads to incomplete pharmacological blockades. These cellular mechanisms are triggered and terminated across 3 distinct phases of a meal. The cephalic phase is anticipatory. Triggered by the thought or smell of food, the vagus nerve initiates acid production before any food is swallowed, accounting for about 30% of the total response. The gastric phase begins when food enters the stomach. Distension and chemical triggers cause a concentrated, reactive gastrin spike that drives roughly 60% of secretion. As the stomach empties, the intestinal phase acts as a feedback brake. As the duodenal pH drops below 3.0, secretin and somatostatin are released. These hormones suppress parietal cell activity and slow gastric emptying. This tightly regulated loop ensures that acid production is titrated to meet the specific mechanical and chemical demands of each meal. In clinical practice, this physiology is critical during anesthetic induction. When airway reflexes are temporarily ablated, the acidity of the stomach presents a severe risk for pulmonary aspiration. Aspiration of fluid with a pH of 1.0 destroys alveolar tissue, resulting in a chemical lung injury known as Mendelssohn syndrome. To manage this, anesthetists use H2 blockers to reduce acid production and sodium citrate to buffer the existing acid already present in the stomach lumen. Mastery of the parietal cell's thermodynamics, receptor synergy, and feedback loops is what allows the anesthetist to manage the stomach's chemical output during the most vulnerable moments of induction.